Hello everyone, I'm Anthony D'Souza and welcome to this series of videos on the exciting world of healthcare science.
I'm Suzanne Crick and I'm a Senior Specialist Biomedical Scientist in the SIH-MDS Haematology Department at Great Ormond Street. So SIH-MDS stands for Specialist Integrated Hematological Malignancy Diagnostic Service. I'm specialized in the molecular hematology section within the SIH-MDS department, and we deal with two main groups of patients. So the first one is children that have been diagnosed with a special type of leukemia, which is acute lymphoblastic leukemia. And these children will receive chemotherapy, or also more difficult cases will receive CAR T-cell therapy. We monitor their cancer levels using molecular methods and to see after the treatment how much residual cancer is still left in their bone marrow. The second group of patients that we deal with is the children that had a bone marrow transplant. At Great Ormond Street, we transplant the patients for a variety of diseases, and that includes leukemia, primary immunodeficiencies, metabolic diseases, and also very rare other types of hematological and immunology diseases. I initially wanted to be an astrophysicist, and I did start to study physics, but then I thought, oh no, let's just drop out of university because I decided it's much better to go traveling. And then I moved to London, and essentially I was always really passionate about science. So I just started to apply for any job that was remotely scientific or based in a laboratory. And that's how I got a part-time job in the National Blood Service, processing fresh donations and producing or separating them into the different components that can be transfused into a patient. And that essentially opened my door into the NHS. So then I got a medical laboratory assistant role in one of the major hospitals in London in a routine hematology and blood transfusion laboratory. And I think things went quite well. So they offered me to go to university and pay for my bachelor degree. So I was actually funded by my hospital. And I did the bachelor degree part-time while still working in the hospital. And because of that, I was able to then move from an MLA into a trainee biomedical scientist position, and then further progress to a band 5 and a band 6. And then I decided to do my master's, and I did a lot of molecular modules during that master's. And then I decided that I would like to work in a molecular lab because I found it very fascinating. I'm generally very interested in research. So I have completed a pre-doctoral fellowship with the NIHR last year, and that went very well. And I've also been accepted for a doctoral fellowship with the NIHR. So that means I will be doing a research project and complete my PhD at UCL. We receive cells after the treatment and we process them. Depending on the patient, the treatment, and the kind of disease they have, we have different means of processing the cells. For bone marrow transplant patients, we tend to sort the cells using a magnetic bead system. This way we can separate individual cell lines such as the T cells, the B cells, and the granulocytes. For the assessment of minimal residual disease, for the leukemic patients, we tend to use a fical separation to extract the cells that we need for analysis. And then we would then extract a DNA from those samples to be able to process them using molecular methods and PCR-based methods. For the bone marrow transplant patients, we perform chimerism analysis, which is genetic fingerprinting. And this way we can tell how much of the recipient cells are left and how much of the donor cells are present after the transplant. One of the new methods we have in the laboratory is the digital droplet PCR. We use that for the patients that have undergone CAR T-cell therapy for acute lymphoblastic leukemia. This involves 3 main steps. There's a digital droplet generator, a PCR machine, and then a droplet reader. And this way we can find out how many copies of the actual CAR T cells are in the patient's circulation, if they are working, and if the cancer is disappearing. We work very closely together with the clinical team based in our laboratory, and also with the bone marrow transplant team. So if we do have any results where there are any changes that might be significant to the patient's treatment, we communicate them straight away. So it's very rewarding to actually see the children recover from very rare and most of the time life-limiting diseases. So I'm happy to be a part of that.